Hello everyone, I'm Satoru Nagatoishi, Project Associate Professor at the University of Tokyo. I'd like to thank this organizer for having an opportunity to present our study at this conference. Today, I'd like to talk about our recent Raman study for biopharmaceutical antibodies. This is a presentation outline. First, I introduce the background of the risk for therapeutic antibodies. Today's presentation, I'd like to discuss three topics for highly concentrated antibody solutions using Raman spectroscopy. In our study, the target protein is antibody. The antibody is an immunological protein which is a specific antigen binder and has a large molecular weight. So, many biopharmaceuticals and therapeutic antibodies have strong potential as drugs against cancer and rheumatoid arthritis due to their high efficacy. Furthermore, in the biopharmaceutical industry, liquid formulation of antibody are becoming increasingly popular. This table is a list of representative therapeutic antibodies. As you can see, the final concentration of antibody solutions are almost high, over 50 mg per ml. So, in the formulation process, highly concentrated, sometimes over 100 mg per ml protein solutions are required. However, a highly concentrated antibody solution has several troubles. At high concentrations, the antibody solution increases its viscosity, so it sometimes becomes difficult to prepare the antibody formulation. In addition, the increased concentration and viscosity is accompanied by an increased risk of protein aggregation or denaturation. These unfavorable changes may result in side effects such as a low drug efficacy and drug toxic reaction. So the optimization of antibody formulations and understanding of the behavior in a highly concentrated solution is of great importance. Now I will talk about the next topic, the physical behaviors of high concentration protein solutions and the analytical standpoint of Raman measurements. Based on the biopharmaceutical background, what kind of behavior we should investigate and understand for highly concentration solutions? This slide shows the difference between diluted solution and highly concentrated solutions. In the diluted solution, the dominant interaction is electrostatic repulsion. So we can study and discuss that protein based on the colloidal approach. However, in a highly concentrated solution, the short-range intermolecular interactions also work. So we have to study and discuss the protein based on the conformational approach. In other words, conformational knowledge is more important in highly concentrated solution. So which instrument, analytical method, is a powerful tool to detect accurately and discuss clearly the conventional behavior of antibodies at high concentrations? This is a table of various analytical methods for detecting colloidal and conventional information. As you can see, Raman spectroscopy is one of the attractive methods to detect and discuss the conventional behavior of proteins at high concentrations. So in this study, we performed Raman spectroscopy to analyze the therapeutic antibody. Now I will talk about the first study, the analysis of IgG solutions at different concentrations using Raman spectroscopy. The study in this presentation was carried out as part of a joint research project with Horiba Ltd. This slide shows the representative Raman bands for protein structure. We can analyze the amido 1 and amido 3 bands, so we can discuss the secondary structure of proteins at high concentrations. In addition, aromatic amino acids, tryptophan, tyrosine, and phenylalanine show unique Raman bands. So we also study the conformational information of proteins at high concentration. This is a Raman spectrum of IgG at 100 mg per ml in the wave number region from 200 to 2000 cm-1. There are many bands for each functional group of protein, and we call a fingerprint region. From this spectrum, we can obtain information about conformational changes and the changes in the microenvironments of the amino acid residues. Especially, we can discuss these 4 types of conformational information: tyrosine, tryptophan, phenylalanine, and amido 1 band. Each amino acid and its chemical structure is identified by each wave number. As shown here, these spectra are the amido 1 bands of each concentration from 10 to 200 mg per ml of IgG. This result shows no specific change in the amido 1 band at each concentration. Therefore, in these IgG concentrations, there is no significant change in the secondary structure of the antibody. When we analyzed the spectrum in detail, several changes in Raman peaks were observed with increasing the IgG solution. This slide is a Raman spectrum of tyrosine residue at 830 and 850 cm-1. Many tyrosine residues are part of the FAB region. Therefore, we expected the 850-830s ratio to be sensitive to tyrosine side chain interactions with neighboring protein molecules. A low ratio value indicates that the OH group of tyrosine acts as a strong hydrogen donor. An increase in the ratio indicates that the OH group acts as both a hydrogen acceptor and hydrogen donor. Our previous study of lysozyme solution has shown that the ratio of these peaks could be used as a tyrosine side chain interaction marker. In the case of our study IgG solution, as you can see in this figure, the ratio starts to increase around 30 mg per ml and becomes saturated at concentration 80 mg per ml. So, this result suggests that the side chain begins to interact around 30 mg per ml concentration and becomes saturated at concentration greater than 80 mg per ml. Okay, I'd like to move the next aromatic residue. This is a Raman spectrum of tryptophan. Tryptophan has an inner ring, so the Raman band for the ring breathing mode of the inner ring appear at 160 cm-1. We found that the width of the band did not change, with concentration in the range from 10 to 200 mg per ml, indicating that the conformation of the inner ring was unaffected by the change in concentration. Tryptophan has another Raman band. The band at 1555 cm-1, and the peak is a commercial marker that provides information about the torsion angle of tryptophan. In contrast to the width of the 760 bands, the width of the 5055 bands increases with increasing concentration up to around 50 mg per ml, as shown here. This result indicates that as the concentration increases, IgG molecule interactions. Next, we analyze the fingerprint region in detail using principal component analysis. We found 3 components, and first and second components are assigned to the stretch vibration of steric acid, which is the buffer element. Interestingly, the third component has a unique profile because the concentration of this component increased at concentrations greater than 80 mg per ml. We identified the spectrum the third component, which is the band intensity of phenylalanine at 1004 cm-1. We plotted the band intensity ratio 1004 1240s because the amide-3 band at 1240 has almost the same intensity. This increase in the relative intensity of phenylalanine indicates that the interaction around phenylalanine changes. Consequently, we concluded that the band intensity ratio 1240s can be used as a marker of the short-range attractive interaction, such as CH-pi interaction. This is a short summary of topic 1. From the analysis of amine 1 band, the secondary structure of IgG did not change, so the structure of IgG is almost stable in the region of the antibody concentration in this study. However, we revealed the conformational change of tyrosine and tryptophan. The side chain began to interact at a concentration of 30 mg/ml and became saturated at concentrations greater than 80 mg/ml. Furthermore, over 80 mg/ml from the band intensity of phenylalanine, the CH-pi interactions increased. So our Raman analysis demonstrates that attractive short-range interactions between IgG molecules began to occur as the concentration increased. Recently, a review of Raman spectral analysis of antibody denaturation and aggregation has been reported. Their findings on tyrosine and tryptophan residues have also been reported. Thus, a detailed analysis of the Raman bands of aromatic rings may be a valuable indicator for analyzing antibody samples with high concentrations. Now we move on to the next topic. In this section, I will discuss Raman analysis of high concentration antibody solution with peak change. Since this study is unpublished data, so specific antibody names and details of the experimental conditions will not be disclosed. Sorry, please understand that the details will be withheld. This slide shows the analysis of Raman peaks of tyrosine and tryptophan with pH change. The spectra on the left result from plotting the intensity ratio 850-830s. First, please look at the black circle. As you can see, the intensity ratio decreased at pH 3. This suggests that the intramolecular interaction of tyrosine is suppressed. The spectra of the right shows the bandwidth, the Raman peaks of tryptophan at 1555. As you can see, the bandwidth also decreased at pH 3, indicating that the conventional repression of the tryptophan band is also lifted at pH 3. Thus, it is suggested that the dispersibility of high concentration antibodies tended to enhance when exposed to acidic conditions such as pH 3. The same Raman measurements at low pH were performed using antibody Y as the other model antibody. As you can see, a decrease in the intensity ratio of the tyrosine band and a decrease in the tryptophan bandwidth were observed at pH 3 of this antibody. As well, when this pH 3 condition antibody sample was neutralized and the neutral condition sample was subjected to Raman analysis, it becomes clear that both the tyrosine and tryptophan bands returned to their pH 7. Please look at the white circle. Thus, the pH 3 condition for high concentration antibodies strongly suggests that dispersion is enhanced and reversible. We then investigated the heat tolerance of high concentration antibodies under the pH 3 condition. The representative Raman bands were analyzed at 1004 for the phenylalanine band, 1555 for the tryptophan band, and 1670 for the amino 1 band. White diamonds are pH 3, white circles are pH 7, and gray circles are acidic treated samples. First, please look at the white circle at pH 7, where a decrease in the intensity of the band was observed around 60 to 70 degrees C. This means that the antibodies are denatured or from aggregates or precipitates. This change was observed in both the tryptophan and amino 1 bands. On the other hand, at pH 3, a completely different change was observed. As seen in the white diamonds, there was also no change in band intensity, even when heated to 19 degrees C. This means the antibodies do not precipitate in the pH 3 environment. In other words, the proteins are dispersed together without aggregation, even at high concentrations in the pH 3 environment. A different change was also observed for the acid-treated antibody sample in the pH 7 sample. As shown in the gray circles, the temperature at the precipitation occurred shifted to a temperature higher than pH 7. This means that the pH 3 exposed high concentration antibodies may have undergone irreversible local conformational changes. Furthermore, this conformational change may be a conformation more resistant to heat stress. To perform more detailed analysis of this heat resistance, we measured the particle size of aggregates using dynamic light scattering (DLS) to evaluate a colloidal's physical property. DLS data were compared for antibody solutions at pH 3, pH 7, and acid-treated conditions at room temperature and heat-treated condition. As you can see, at pH 3, a single particle distribution was observed around 10 nanometer. Therefore, pH 3 is colloidally stable for the antibody. On the other hand, at pH 7, large particles were observed after heating treatment. Also, aggregates were found to exist upon heating for acid-treated condition antibodies, as in the case of pH 7. Thus, the DLS measurement showed the pH 3 is an environment that enhances colloidal stability. On the other hand, the acid-treated antibodies could not be characterized using DLS. Therefore, we performed particle distribution analysis using particle tracking analysis (PTA). We compared heated and unheated samples and performed PTA analysis on each sample to count the number of aggregation particles. As you can see, only the same number of particles were observed in the pH 3 sample as in the unheated sample, despite being heated. Similar to the Raman and DLS measurements, the pH 3 sample showed that the aggregation of antibody was significantly suppressed. On the other hand, under the pH 7 condition, a significant increase in aggregates was observed in the heated samples. A further interesting result was observed in the acid-heated samples. Despite the pH 7 environment, no significant increase in aggregates was observed in the heated samples. This suggests that the irreversible conformation of antibodies exposed to pH 3 may be resistant to aggregation. Now we move on to the last topic. We have attempted to see if we can track the characteristics of each of the three monoclonal antibodies using Raman spectra. Since these data will also be unpublished data, we will not disclose the specific names of the antibodies and refer to them as antibody A, antibody B, antibody C. Sorry for your inconvenience. The Raman bands, the three antibodies at 50 mg per ml are compared. These profiles are a comparison in the region above 1000. Some of the bands have different shapes, but we focused on the tryptophan band at 1555, was analyzed in detail from the previous Raman analysis. This is the result of observing the bandwidth of the tryptophan Raman bands at 1555 as the antibody concentration change. As shown in this plot, the bandwidth of antibody B is clearly different from that of antibodies A and C, and shows a larger value. This suggests that the intermolecular interaction may be stronger in antibody B. We then compared the Raman peaks in the region below 1000. Tryptophan bands are also present in this region around 760 and 880, and we performed a detailed analysis of these spectra as well. Here are the results of the observed tryptophan Raman band intensities of 760, 880s at different antibody concentration. In this plot, the intensity ratio of antibody B is clearly higher than that of antibodies A and C. This also suggests that the intermolecular interaction may be stronger for antibody B. So what is responsible for this difference in Raman bands among different antibodies? We performed light scattering SLS measurements at different antibody concentrations to analyze the second Billiard coefficient. Here is the result of plotting the Lowry ratio divided by the optical constants against the antibody concentration change. As you can see, a positive slope was observed for antibody A and antibody C. A positive slope means that the proteins tend to cause repulsion from each other. Therefore, antibody B, which has a slower slope, is considered less repulsive than the other antibodies. We also calculated the D22 value of the Kirkwood-Buff integrals for each antibody. As a result, a profile converging from positive value to 0 was observed for antibody A and antibody C as their concentration increased. However, a change from negative value to 0 was observed for antibody B. This result suggests that the properties of antibody B are different from those of other antibodies. This change in D22 value may mean that the antibody B is more likely to interact with each other at high concentrations. Thus, we obtained results from Raman measurements between different antibodies that satisfying a new indicator to identify the different interaction behaviors among different antibodies. So this is our conclusion and summary slide. In this presentation, we used Raman spectroscopy to evaluate the highly concentration antibody solution. Especially in the different antibody analysis, Raman bands may be able to detect differences in the intermolecular interactions among different antibodies. This is acknowledgement in our study. I want to say many thanks to Professor Tsumoto and its lab staff members and the staff of Horiba Company for supporting and advising this study. And this research was partially supported by Japan Agency for Medical Research and Development, AMED. Finally, I'd like to say that I believe Raman spectroscopy is a bridge to success in the biopharmaceutical formulation. Thank you very much for your kind listening.